So today is Lab 5. A little bit about logic, if-then statements, basically hypotheses. And then we'll go into the two stains that you're going to do today, gram stain and capsule stain. So how do you make and test a hypothesis? We typically use if-then statements. A hypothesis is if this, then this, right? And you test hypotheses from the time that you're a baby, right? You drop your bottle, say, on the floor. Every time it falls and hits because we now know about gravity, right? But, you know, you got to test it, right, to know if it happens. If you cry, right, what is mom going to do, right? We use logic all the time. We test hypotheses all the time. If A, then B, right? If I do this, what's going to happen? Or we can even assume, based on prior knowledge, this should happen, right? And we could test it. So, one morphology, right? One shape, one color, say on a petri dish, is just one species. Is that true? No, right? Right? As we've learned, right, and as I have said, and unfortunately I don't have my plates because I don't have a place to store them. But one of the mixed cultures you guys had was Staphylococcus and Klebsiella. They're both white, right? So even if you've got isolated colonies of two different ones, it's really hard to tell them one from the other, right? At that level, okay? Under the microscope, one's a cocci. No, actually, they're both rods. So again, in that case, you wouldn't be able to tell the difference. You know, no, one's a cocci. Klebsiella is a rod. Staph is a cocci. Staphylococcus, right, is a cocci. So morphology-wise, you would be able to tell the difference. But there are some, you know, they're both rods or they're both cocci. You're not going to be able to tell the difference. You have to run additional tests, right? So another way of writing things Peter refers to as upside down and backwards, but is to kind of flip it, right? So another way you could say one morphology equals one species. You could also say two or greater species would equal two or greater morphologies. Sometimes it's true, not always. As I said, the example with Klebsiella and Staph. They're both white, right? When you grow them on the petri dish, you're going to get just one morphology. So the second part of this statement is false. Everyone still with me? In order for our hypotheses, right, we've got to have both statements be true, both parts of it, right, or both parts be false, right, in order to accept a hypothesis. So if A and B are true, but A equals B is false, right, we're good. But we can't have A be false and B be true. Okay? So in this case, right, we have greater than two species. We've got Staph and Klebsiella, right? But we only had one morphology. So this part of the statement is false. Still with me? Okay. So a hypothesis is disproved if the first part of the statement is true while the second part is false. Right? This is not an accepted hypothesis. Observing one morphology of a mixed culture would disprove, right? Because it's mixed, it's more than one species. Right? So we disprove this hypothesis. If a hypothesis cannot be refuted, then we have supporting evidence for it. And some hypotheses have been so supported throughout the years that they're referred to nowadays as theories, right? Not to say that somebody one day might not disprove it, right? I'm going to throw out that theory. So this is one of the ways of writing. It's a mathematical way. Remember, sometimes I said I present information in a lot of different ways, right, in hopes of getting everybody to understand it. So in English we would say it's necessary and sufficient, right? For A, you have to have B occur, right? So again, these two have to be linked up. We cannot have them contradict each other, right? They either both have to be true or they both have to be false. We can't have one be true and the other be false, right? They've got to be linked up to each other. So later on in the semester, we're going to do a pretty cool experiment. We're going to do bacterial transformation. We're going to take DNA that's originally from jellyfish, and we're going to put it into E. coli. This DNA can be made into a protein that fluoresces green under UV light. This is referred to as transformation, right? When they take up the DNA and they utilize it and they transform, they can do something they couldn't do in the past. They can only do this, they can only fluoresce green if they actually take the DNA and they express it, right? Go from DNA to messenger RNA and actually make that protein, right? So these two things are linked. You cannot have expression and not fluoresce. And you cannot fluoresce without expressing that genetic information. Make sense? Right? So those two are linked, right? So you've got to have fluorescence. If you have fluorescence, you've got to have expression and vice versa. They're not moving along, right? Go away, noisy people. Might have to say something to them. Okay. So, as I said, even from young children, right, we test hypotheses all the time. Lots of different professions are professionals at that, right? Always trying to prove and disprove statements, right? Always testing hypotheses, especially lawyers, right? They're always arguing stuff. Doctors and such, right? We use process of elimination in trying to figure out and diagnose and treat patients. Engineers, mechanics, plumbers, information technology, and the list goes on, right? And ourselves, right? You use this process of testing a hypothesis all the time. You may have formerly been introduced to it as the scientific method, right? Right? We form a question, a hypothesis. If this, then this, right? So, journal articles, scientific articles are always set up this way, right? You have an introduction where they present data, right, and present their hypotheses, their if-then statement, A, then B. So, why am I doing these experiments, right? Because I think this will happen. Methods, right? How do I do this, right? So, we're going to be widely used and approved methods, right? Protocols, right? That's what those methods are called, procedures. We have to do them correctly, right? You have to come prepared to class as you guys have. Write out your procedure, right? Follow your procedure. If you don't follow your procedure, what should you do? Document that you screwed up, right? And if you were doing an experiment, you would have to what? Start over because you did not follow the protocol, right, that you meant to follow. Okay. For in this lab, right, sometimes we'll have to repeat, especially when you're trying to figure out your unknown. For these, right, you're just learning. If you mess up, just document, right? Where you went wrong, look at someone else's slide, right? So, how do I perform these experiments properly, right? Results. What are our observations, right? Commonly, we're going to draw things as you saw, right? Trying to get you guys to be good recorders of your evidence. Taking pictures is fine too, and we did that last time, right? Drawing, documenting, writing. Sometimes they'll present information, results in datas or graphs, right? But the information is going to be there, right? Our observations of what we saw, right? Unless you've accidentally discovered something new, you're going to, of course, eliminate when you know you've made a mistake, right? When you know you didn't do something correctly, you can't you can't write down those results, right? They would screw up your experiment. So to give you a real example, like when you do the gram stain, if you realize you forgot the iodine step, can you rely on your results? No. You know it's not come out accurately. That's an important step in the process. You can't skip steps, right? So what outcomes should I record, and how should I report them? And then in the discussion, we justify any ambiguous or contradictory results, which you may have to do for your unknowns, and we draw conclusions, right? So what does this mean, right? What does this observation, what does this result mean, right? So the gram stain that we do is actually named after a gentleman who came up with this procedure, right? And is widely used in microbiology because it helps us distinguish different types of bacteria. You can use it to stain other types of cells, right? But mainly we do this for bacteria because of their cell wall composition and how it interacts with the dyes will give us the two different results, whether it's what we call gram-positive result or negative. And again, this is a result. It has nothing to do with charge, right? So we're not saying the cells are positive or negative charge because all of them are always what charge? Negative. Remember, bacteria, all cells in general are negatively charged because of their membranes, right, and their DNA is highly negatively charged. Which is why we use a lot of basic dyes, right, because they're positively charged and they're attracted to those negative components. Go ahead, Andrew. Nope, you're one step ahead.
So
we're getting to it.
How would he? What kind of hypothesis would Graham use to arrive at the technique? It seems like out of the blue, he discovered that.
Right. So he was playing around with different dyes potentially, and he discovered that he ended up with two different results. And so then the question became why, right? Why when I do this I get this? Right? And so scientists since then, right, have deduced down to the chemical level, like Andrew said, this substance that's unique to bacteria that makes up their cell walls called peptidoglycan determines these differences that we get when we run this reaction. So gram-positive, as I said, doesn't mean that they're positively charged. It means that the result in this test is that they stay purple, right? They start out purple and they stay purple. And the reasoning behind that is these cells that stay purple have a very thick layer of peptidoglycan. This is the substance that makes up the cell wall of bacteria. This is unique to bacteria. And in addition, they have tectoic, I'm going to say it wrong. Tectoic, thank you, acids. And they're represented here by these squiggly lines. The cell wall itself, even gram-negatives, tend to have a very thin layer of peptidoglycan, this cell wall substance, for bacteria. This helps them hold the different shapes that we looked at last time, and we'll still see shapes again today, right? And it also helps them with osmotic pressure. Most of these cells have more water outside of them than inside. So water tends to come into these cells, and if it wasn't for the cell wall, they would burst, right? So this helps them resist that bursting. It helps maintain and keep them intact, as well as they do other things to help with that osmotic pressure, that pressure of water rushing into their cells. So for gram-negative cells, they're going to become decolorized, and then they're going to stain with the counter stain and come out pink. And the reason for that is this thinner layer of peptidoglycan they typically have. And they typically also have an outer membrane. So this is an additional membrane to the plasma or cell membrane that all cells have. Okay? And of course it's the outermost covering in this case. And its very outer layer is made of a different substance than phospholipids. It's actually lipopolysaccharides. The bad news for us, this can be very toxic, and this is why gram-negative infections can be really serious. So there's my rebar. So these guys are almost like supporting, and the reason for that is that very thick layer, lots of layers of peptidoglycan that usually are present for gram-positives. That's why they also have the tectoic acid. It's almost like rebar, helps support it. So when we're staining these guys, you're not going to actually see this layer. right? This layer, especially for gram-positives, which can be really thick, up to like 30 layers of peptidoglycan, measurement-wise, it can range from 30 to 100 nanometers in diameter. What's the resolution of our microscopes at oil immersion? You remember? 200 nanometers. 200. Right? So this is too small, right? This is not within our limit of resolution to be able to see. So it wasn't until the advent of things like electron microscopes, right, we could see these things in greater detail, see these additional layers, see these different thicknesses. So peptidoglycan, right, also known as murine, it's made up of two sugars, N-acetyl uric acid and N-acetylglucosamine. These alternate, and this creates a mesh over the entire surface of the three-dimensional cell. Each layer is then hooked together by amino acids, right? And especially for gram-positives, because they tend to have a lot of layers, they have an additional set of amino acids that cross-link the first set of amino acids. So layers upon layers, imagine like a jawbreaker, right? Layer upon layer of that candy, layer upon layer of the sugars linked with the amino acids. This is a very rigid structure, but it's very porous. Stuff can go in and out of it very easily. And then, of course, our cells heat fix our slides. What's the reason why we heat fix? They shrink, but that's not why we do it.
And allow them to adhere to the slide.
To get them to stick to the slide, right? To get them to stick. But what else happens? It kills them, right? So remember, these aren't living cells, so they can't, their membrane can't resist, right? These dyes are going to go rushing in, okay? And the membrane's not going to stop them. Other types of substances that may be associated with organisms on the outermost covering, like bacteria, are glycoproteins. These are proteins associated with sugars. And so these interact a little bit differently. So notice these sugars here and their particular binding, right? Where these are alpha long strings of amino acids that form these alpha helixes, and here are the sugars. This is a very common situation of having protein and sugar connected to each other in this way for proteins and stuff that the cells may secrete, or even things that the cell brings in. So this is that other animation, right, that you guys also have access to. And I'm not going to play this one. Instead, I'm going to show you this other one, which is listed as gram stain animation 2. It doesn't have any audio, but I really like the graphics. So for gram stain, we have two possible results. Positive meaning it stays purple through the whole process. Negative meaning it decolorizes and we stain it with a counter stain and it comes out pink. Shape doesn't determine, right, whether they're going to be pink or purple. It's the cell wall composition, how many layers of that peptidoglycan they have, that determines whether they'll stay purple or not. So we're going to start out with the gram positives. So as you can see here, this layer is meant to represent the peptidoglycan layer in the animation. This is meant to represent the tectoic acids. No idea why. There's our phospholipids, proteins embedded in the membrane, and then the different dyes and dye complexes that we'll use, right? So C is for crystal violet, D is for the decolorizer, I for iodine, S for safranin, and then CI for when we form that crystal violet iodine complex. So and they even have it written out here, right? But I'm going to pretty much say what it says here. So the cell, for a gram-positive cell, if you could look at it in this detail, it's going to have its cell membrane, and it's going to have a layer peptidoglycan with the tectoic acids associated with it. The first step would be to add the crystal violet. The crystal violet, remember, is a basic dye, so it's going to go into the cells. It's attracted to the negative components of these phosphates, as well as the DNA inside the cell. So it travels in, right? Goes through those layers, goes into the cell. The next step is adding Graham's iodine. For your guys' procedure today in the book, it says Graham's reagent. It is actually iodine. Okay, so make sure you make a note of that. So then we apply the iodine. It is going to go into the cell. Crystal violet has a chlorine ion associated with it that's negatively charged. These two switch out with each other. So now it becomes crystal violet complex with iodine instead of chlorine. So they complex together. This makes it a larger molecule than it was previously with the chlorine ions. So when we do the next step, which is the decolorization, the way the ethanol affects this peptidoglycan layer is it typically causes it to shrink up. And so you'll notice the space between the molecules gets much smaller. And this essentially will trap in that larger crystal violet iodine complex. So notice it's not going to come out the cells, right? It gets trapped in. So they stay purple. The next step is mainly for our gram negatives, but even our gram positives will get stained with safranin, which is a red dye. It's another basic dye, so it's attracted to the inside of these cells. It's smaller, right, so it can fit through. But what color are these cells going to appear? They're still going to appear purple even though you stain them with the red dye, because the purple is a much brighter color. They'll even look a darker purple, actually, than if you didn't do this step, okay, because of the addition of the red dye. So under the microscope, they're going to look purple, which we record as gram-positive result, meaning they stayed purple. Now, for the gram-negatives, they have a different architecture, right, to their cell wall or their cellular envelope, including this outer membrane. So they have their normal cell membrane. They tend to have a very thin layer of peptidoglycan, and they have an outer membrane. And again, that outer membrane, the outermost section contains a little bit different substance. It's not a phospholipid, it's lipopolysaccharide. So it's lipids with these sugars associated with it. And the bad news for us, as I said, that this could be quite toxic for humans. So same steps, right? Crystal violet goes into the cell because it's attracted to those components. So the cells are going to be purple at this step. We're going to add the iodine. It will go in. It'll complex with the crystal violet, make it a larger molecule. Except this is the different step here, right? What makes this a differential stain, why you end up with two different results, is that when you decolorize, even though it shrinks this peptidoglycan layer because it's so small, it's not enough to trap in the crystal violet iodine. See how it still gets out? Also, what disappeared? The outer membrane. Because membranes, lipids, can be dissolved by ethanol or things like acetone. We don't use acetone though. It's damaging to mucous membranes. It's yucky stuff. Alcohol works great as a decolorizer though. It'll even remove permanent marker unless the permanent marker says alcohol resistant. Alright, so permanent isn't really permanent. It just depends on what it's on though. Okay? So the crystal violet iodine gets washed out. So at this point, if you were to stop here, these cells would be what?
Colorless.
They would be colorless. But we want to actually be able to see them, right? So then we have to counterstain them with safranin, which is a red dye. Again, basic, positively charged, goes into the cells. It's not going to be able to retain a lot of this dye because of the damage that it incurred during decolorization. So they're not going to be red, but they'll appear pink under the microscope. So there we have our pink, which indicates the result that we know as gram-negative. Okay? So back to my PowerPoint. So Ah, that's the answer. Okay. So this is a toy right here, this one. Don't I have it? So yeah, there's that one. That most kids had when they were little. And it has two parts, right? You can separate the two parts by grabbing the handle at the top and the bottom, and the little blocks fall out. Most little kids don't know this, though. And these holes, as you can see, are different shapes, and those blocks just fit in those holes perfectly. So most kids fill up the whole thing, right? And then they want to do it again. So then they stick their little hand in the hole, and they try to grab onto the block. But the little hand and the block together are not going to fit back out that hole, right? Those two things combined are not going to get back out that hole. So this is very similar as to what happens and why that iodine step is so important, is crystal violet goes in, just like the block goes in, right? Then the iodine goes in, just like the hand goes in, and the two combine. So now they're larger, and so it's harder to get them out. And then, for gram staining, what also happens to that hole when you decolorize? It shrinks. Can you imagine sticking your hand in and the hole shrinking up on you? You'd really have a screaming kid, right? So every time you got to convince the kid, right, the first time this happens, to drop the block out of their cute little hand so you can get their little hand back out the hole, right, and teach them how to open it, right, so that they can get the blocks out and do it again. All right, so this is your quiz for today. Those of you guys in the lecture don't get to answer it because you've already seen it, right? This is an intelligence quiz. For this picture, right, how do you get this ball out this hole? What do you have to do? Take the pegs out, right? See the little pegs? You got to take them out, otherwise it's not going to fit, right? It's going to block it. How did they get it in there to begin with? Those two things went in separately. Ball, then pegs. Make sense? All right. Okay. So the same thing with crystal violet iodine. They go in separately, right? But then they combine. And because they combine, they're larger and it's more difficult to get them out. In addition, that crystal violet, additionally, the peptidoglycan layer shrinks, right? The holes shrink up. So why is it bigger? Well, it's bigger because crystal violet is this, and it's complex with the chlorine ion, but we do a switch for iodine when we add the iodine. They're both negatively charged, right? They're in the same column of the periodic table. The difference is iodine is bigger. It's a bigger molecule, right? So this is Peter's supposed stray cat. He's had her for a while, so I wouldn't consider her stray, right? But she adopted him. And you can see her squeezing through his fence there. Guess what happens after he feeds her? She can't fit back through the fence, right? She can't leave for a while, right? So little small changes, right, make a big difference on how molecules interact in this cute little kitty cat, right? So switching out that chlorine for the iodine is about 123% bigger, right, as the same thing happens with the cute little kitty, right? Sometimes small differences can make a big difference. So each one of the steps, right, gram positive, right, they're purple, right, it's a basic dye, it goes in, it stains all the cells purple. Iodine's not going to affect the color, right, at the molecular level, although I'll tell you, even on my slides, you'll notice it'll kind of turn black, and this is because iodine, when it interacts with sugars, like starch, it'll turn black, right? If you put it on a potato, it'll turn black. That's also kind of a good indicator that you didn't forget that step. When you decolorize, these guys, if you do it correctly, should stay purple, right? Your gram positives. Again, because we're shrinking it in and trapping in the crystal violet iodine. And then we will stain them with safranin, but remember, it's going to stay and appear purple because the purple is brighter than the red. For gram negatives, right, we're going to stain them purple. We're going to add the iodine. The difference here is at that decolorization step, right? We're going to damage or even remove that outer membrane. The peptidoglycan layer is not going to be able to shrink up enough. It's not thick enough to trap in the crystal violet iodine. It becomes colorless at this step if you were to stop. So in order to see them, you must counterstain, right, with safranin in order to see these cells, and they should appear pink. So again, all of them get stained purple. All of them get the iodine and get the complex. The difference for this stain is that decolorization step, right? This is where you're going to get the differences because of the differences in the cell wall composition and how much is there. And then we stain with safranin. So another way of looking at it, right? All of them are going to be purple. All of them get the iodine, right? We decolorize. Only the gram-negatives should lose the color. Then we stain with safranin. As I said, you're going to actually get a deeper purple. And then you're going to get a light red or pink for your gram-negatives, if done properly. So what are some mistakes you can make, and why is it important not to do them? Well, because you will not be able to distinguish one from the other if you skip or don't do any of these steps or don't do them in the right order. So we'll look at each one of these mistakes independently, but let's review what are the 4 major steps. Number 1, crystal violet. Number 2, iodine. 1 minute, right?
Yes.
Decolorize with ethanol, alcohol, which is an alcohol. Do not grab the acid alcohol, right? That's for next week. Right? Make sure it's the ethanol. We're going to do this drop-wise until the drops coming off go from a deep purple to a lighter purple. And I am going to demonstrate this, right? Because this is the crucial step. If you decolorize too long, you can even remove it from your gram-positives. And then safranin so we can see our gram-negatives, right? 1 minute. Our secondary or counter stain. So if you take those into consideration, what happens, right, as we have at the molecular level, if you skip the iodine step, right, are your gram-positives going to come out purple like they should? No, why not?
They'll be able to stay.
Because the crystal violet is going to be smaller, right? And because it doesn't have the iodine, so it's going to wash out, right? So purple step, fine. Omitting iodine, not good, right? We're not going to get that complex, not getting that larger molecule. It's going to decolorize, right? So then we're going to stain them with safranin. They're going to be pink. Gram-positive means purple, though, right? So is this the correct result? No.
Yes, not.
What happens if we do this very same thing and we forget the iodine for our gram-negatives?
They stay pink as
Yeah, they're going to come out as usual, right? Because they always get decolorized at the decolorization step. So even though we forgot the iodine, it's no big deal for gram-negatives, except if you made this mistake, how are you going to distinguish gram-positives from gram-negatives? They're both what? Pink. You can't. Right? So this step, right, is crucial to getting the correct result. If you know you forgot it, you cannot trust your results. Okay, what happens if you don't decolorize? Are your gram-positives going to come out as they should?
yes.
Yeah, because they don't get decolorized anyways, right? So we're still going to, but it's going to be a lighter purple. I mean, no, they're still going to be purple. It's going to be fine. Thinking ahead. It's when we do the gram-negatives, if we don't decolorize them, right, they're never going to become colorless to then become red. Instead, they're going to stay purple, which is the same as our gram-positives, which, again, we got 2 purple cells. We can't tell the difference between gram-positive and gram-negative. What if you forget to do the safranin? For gram-positives, is this going to matter?
No.
No, you're just going to end up with a lighter purple. No big deal. Right? What about your gram-negatives? They're going to be colorless. You're not even going to see them, right?
Yes.
The reason why we use the safranin is to see them. Right? So again, this would be bad because you wouldn't even be able to see them. This one I've actually done just to see how it would come out. I've actually reversed and done the safranin first and the crystal violet as the opposite. So for gram positives, right, what's going to happen is they're going to get stained red, right? And safranin does probably complex with the iodine. And probably, again, makes a larger molecule than previously. Gets trapped in when we decolorize. We stain with crystal violet. They're going to be a nice deep purple, like we're used to seeing, right? So yes, no problem here. The problem is with our gram negatives. So if you think about this process, we stain them with safranin, right? We add the iodine, it complexes. What happens when we decolorize? The red is going to be removed, right? And they're going to be colorless. So then now we're going to stain them what? Purple. Oops. What color are the gram positives? Purple. A darker purple. But do you want to try and determine light purple from dark purple?
No.
No. That would be like next to impossible. And the only reason why I could see this difference is I did a mixture, right, of gram positives and gram negatives next to each other, one a cocci, one a rod, so I knew which one was supposed to be gram positive or gram negative. And in fact, the cocci, which were supposed to be gram positives, were a deeper purple than the purple rods, which were supposed to be gram negative. Okay? So there's a reason why we do it in the order that we do, right? So it's easy to distinguish the two types. So both of these are basic stains, crystal violet and iodine, right? I mean, safranin. They're both going to stain the cells. They're positively charged. But why is it that when we use them in this procedure, we can distinguish between gram positive and gram negative? Because of the decolorization step, right? Because we are using these guys, right? But we're using them in a procedure where, as we said, all the steps matter, even that iodine. Right? So when we do simple staining, we're just using one stain. In this case, we're using multiple stains and we're decolorizing. And we're using the mordant. So that's why you're going to get these different results. So if we were to stain eukaryotic cells, our own cells, first of all, do we have cell walls? No. And what's the special substance that differentiates the bacteria for the gram stain? What's their cell wall made out of? Peptidoglycan. Well, since we don't have cell walls and we don't have peptidoglycan, can we trap in the crystal violet iodine? No, we cannot. So therefore it gets washed out, and so our cells will be what?
Red.
They'd be pink. They would be pink or reddish, right? And so as you can see in this picture, right, that's a cheek cell, the big pink, and then the nucleus, right, which we know eukaryotes have, is colored. These little purple things are gram-positive bacteria, right? So normal people, that's kind of a joke, right? Everyone, right? All of our cells would come out gram-negative if you were to gram stain them. Okay. Even bacteria, there's a group of bacteria that don't have cell walls. What would they come out? If we completed the gram stain all the way through, they would be pink, right? No cell wall, no trapping in crystal violet iodine, right? They won't stay purple. They'll end up decolorizing and then staining with the safranin, so they'll be pink. So, how does scientific method apply? Right? Why am I doing this experiment? Well, we want to know what's the cell wall structure of our bacteria, right? Does it have that thick layer or a thin layer? Does it have the outer membrane probably? Right? How do we perform it? Right? We got to do our steps and do them accurately. How do we record? Well, we know if it's purple, it's gram positive, right? If it's gram negative, it'll be pink. We'll draw this, we'll write this in words, we may even take pictures, right? What does it mean though? Right? Gram positive being purple means it has that thick peptidoglycan layer that traps in the crystal violet iodine. 40% of the known bacteria are gram positive. The other 60% are gram negatives, right? For the most part, they're going to have that thin peptidoglycan layer and an outer membrane. There's a few, right, that have no cell wall at all. So the next one is our Glycocalyx, also known as a capsule or slime layer. So literally means sugar coat. So as you can imagine, chemical composition is most commonly sugar. Some of them have glycoproteins, right? Sugar proteins. And there's at least one case where it's exclusively just made out of amino acids out of protein. The difference between a capsule layer and a slime layer, right, denoted as the outermost layer for any cell that has this structure. Not all cells do, right? So it would be outside the cell membrane, the cell wall, right? The outer membrane, if it has it, it would be the outermost covering of the cell. The capsules themselves are typically, as I said, polysaccharides, typically sugars, and they're attached firmly to the cell wall, where slime layers are diffuse and not firmly attached, and this, cells can even use them to glide, right, for motility. We're looking for capsule layers. The reason for that is that they aid in attachment to surfaces. So, for instance, attachment to our cells and to us. And it helps them in evading our immune system. It protects them from us. It's almost like they're wearing a disguise. It's really hard for our white blood cells to identify them and even some of our antibodies to find them and destroy them, right? So most of the pathogenic organisms, right, have this structure, have a capsule structure. So Klebsiella pneumoniae, right, can cause a really serious form of pneumonia. Part of the reason is it's got a capsule, right? It can stick to our lungs. It can evade phagocytosis by our white blood cells, right? So it's bad news in that case. Again, no one's licking Petri dishes, right? We're using proper aseptic technique. We're not exposing us to large numbers of bacteria. No one's going to get pneumonia. So for this procedure, we use a drop of Congo Red near the end of our slide, right? Then we're going to add our bacteria to it, and then we're going to spread it out, and then we're going to let it air dry. No heat fixing, right? No heat fixing. Then we're going to flood the side after it's dried on our light boxes for 1 ml reagent. This is going to change the color of the slide from the red, from the Congo Red that we're used to seeing, to a grayish blue. And this is partly due to a pH change that happens. So you'll notice there's acetic acid, which is the same thing as vinegar, in this reagent, so it'll kind of smell vinegary, and that's why. I tried this with my class yesterday. We're actually not going to rinse these slides. Just go ahead and drip off the excess and blot them in your blotting paper. When you rinse, because we haven't heat fixed these slides, a lot of times the pieces sometimes will come undone and wash off the slide, which usually isn't a problem because there's usually enough pieces that stay stuck that you can see. But I found we got much better results if we didn't rinse these slides, right? So just drip it, right? Try and get most of the excess off on your staining tray, and then just put it in your blotting paper, right? And then it's ready to look at under the microscope. And of course, always to oil immersion. So remember, we're kind of doing a negative stain like last time, but then kind of doing like a simple stain where we flood the slide. And because of that, we're going to be able to visualize a capsule because of how those two different stains work, how a negative stain works versus simple staining with a basic dye. So the other thing in the procedure that we do not do is we don't use the serum. But why do they say they use serum? To help it stick, right? And again, because we do not heat fix these slides in this procedure. And the reason for that is you could distort that capsule layer. Because remember, heating, right, will shrink and affect things. So some oral bacteria, some of the ones in your mouth, again, the ones that attach to your teeth, do that because they're encapsulated, right? It has to do with their functionality, right? So what benefit is that? It allows them to attach, and also, as I said, it helps protect them from our immune system, right? Our phagocytic cells that would normally grab on and engulf them can't recognize them. Our antibodies that would tag them for destruction, some of them can't bind to that layer. So this is a little bit different capsule stain than what we do. The colors are different, but same principle. We're going to stain, we'll have the background stained in the cells, and the capsule does not stain. It's a clear halo around the outside of the cell. So ours is going to look more like this, right? That grayish, bluish color. The cells themselves are going to be purplish to pinkish in color, almost like what we call fuchsia, a purple-pink color. And then if the capsule's present, it's going to be this clear, unstained halo around the outside of the cell. What shape is this cell? Rod-shaped, or bacillus, right? So why are we using the Congo red? Well, it's an acidic dye. What charge does it have?
Negative.
Negative. So what does it stain?
The background.
The background, right? That's only the first step. Do not let anybody look at a slide today that's red under the microscope, because once you put that oil on, it's ruined and you got to start over. Okay? You're not going to see the capsule if you stop there. Right? We've got to use the basic stain, the Medville stain. This is positively charged. It's going to stain the cell, right? And it will interact with the background, so the whole slide will change color, right? It'll be this kind of grayish blue color. So, is the cell stained? I mean, is the capsule stained? Sorry. No, right? The cell and the background are, right? The capsule is going to appear clear around the outside of the cell. So, in this picture, do these cells have a capsule? No. No. You see the purple cells? You see the background? Do you see any clear space in between those two? No. Micrococcus luteus should look like this under the microscope. Staphylococcus, that we'll look at today as well. This is another cocci, but this one does form a capsule, but these cells are so small, sometimes it's really hard to see the halos around them. Maybe we'll get a good slide today. Okay? For Klebsiella pneumoniae, y'all, you won't miss the capsule. It's huge, right? It's huge. And then your yeasts are really large too, right? Because they're eukaryotic and they have a nice big capsule. So this species of bacteria does not have a capsule. So why do we have to do a simple stain and a negative stain combined? Because remember, the simple stain is only going to stain what? Simple stain with a basic dye. It's only going to stain the cell. The only reason why you would think this one has a capsule is because I drew a line around it. Otherwise, this would just blend into the background, right? Because the capsule does not stain. So the same is true if we just did a negative stain, right? You're just going to see a bigger cell. You're not going to know that it's bigger because it has a capsule layer. Now I found, and I don't know if you guys noticed, when we did the staining with the Congo red last time, that sometimes it stained the cell. So if you look inside some of those clear spaces, you could actually see a cell, right? Because we use Klebsiella pneumoniae, which you're going to use again today, but today, for sure, you will stain that cell inside, and you'll see that nice big capsule layer, right? So we wouldn't be able to distinguish. So why are we doing? I'm going to answer in a minute, Hunter. I'm not going to forget about you. So why are we doing it? Well, we want to know if they have a capsule or not, right? How do we perform it? You got to do all steps, right? You got to do your Congo red. You got to let it dry. You got to do your Medville's reagent, blot it dry, and look at it. We're going to record our results, right? If it has a capsule, it's going to have that clear halo around the outside of the cell between the cell and the background. If it doesn't, right, no clear halo. You're just going to see cell and background. No space. What's the significance? Right? Because things like capsules or those types of organisms tend to be very pathogenic because that capsule aids in attachment, right? As well as it aids in protection of keeping our immune system from identifying it, from being able to gobble it up by phagocytosis or antibodies binding to it. What's your question, Hunter?
I'm confused that the basic stain doesn't stain the capsule because it doesn't penetrate, but it does penetrate.
No, it has to do with charges. The capsule's not charged, and so the dyes aren't attracted to it. Yeah, that's why it doesn't stain. So it doesn't matter whether we use an acidic stain or a basic stain. it's not going to stain. But we have to be able to stain the cell in the background to see the unstained capsule because it's outside of the cell. Excellent question. Okay? All right. So, oh yeah. And negative ones, of course, don't have a capsule, right? And usually aren't pathogenic. So then I have my glossary of terms for you guys, right? Which I'm not going to go over, but it's there. This PowerPoint is available now, and the recording will be available later.